Chapter 5, the cell cycle. The ability of organisms to produce more of their own kind is one of the characteristics that best distinguishes living things from non-living matter. Key concept: cells have distinct phases of growth, reproduction, and normal functions. The continuity of life is based on the reproduction of cells or cell division. Cell division functions include growth, repair, reproduction. In a multicellular organism, cell division functions to repair and renew cells that die from normal wear and tear or accidents. Cell division is an important part of the cell cycle, which is the life of a cell from its origin and the division of a parent cell until its own division into two daughter cells. We use the IPMAT mnemonic device to help you remember the stages of the cell cycle. To answer your burning question, it stands for interphase, prophase, metaphase, anaphase, and telophase. And of course we have biology. It's the only science where multiplication and division mean the same thing. There are three main stages to the cell cycle. There's interphase, there's mitosis or meiosis, and finally cytokinesis. The cell cycle is a regular pattern of growth, DNA replication, and cell division. We can see here, we'll do this in our notes as well, but there's lots of different stages to cell division. Interphase takes up the majority of a life cycle of a cell, and then mitosis, and finally cytokinesis. The two main stages of the cell cycle are gap 1, synthesis, gap 2, and mitosis. Gap 1, or G1, is cells growing and carrying out their normal functions, so just living their day-to-day life. They're also going to replicate their organelles during this time, because if a cell is going to make a copy of itself, it has to make a copy of all of its insides as well. The next stage, once we reach the end of gap 1, before we move on, there's going to be a little chemical signal stoplight that double checks to make sure that all the organelles have replicated faithfully, that everything is growing and progressing nicely, and then we can enter this DNA synthesis or S phase. The S phase is when the cell will copy its DNA. So this entire chunk of the lifespan of the cell is going to be making a copy of the DNA inside the cell. And it's pretty amazing to think that all of your cells have to faithfully replicate every single nucleotide in your entire strand of DNA. You're copying it word for word. And again, we have another stoplight, so there's going to be another checkpoint to make sure that the DNA copied correctly. If we get a green light there, then we'll enter the G2 or gap 2. During gap 2, there's some additional growth. Mainly what's happening here is the cytoplasm is doubling in size. So that's really the last thing that we need to fill out this cell before we start dividing. And then finally, we enter the M phase, or mitosis. Mitosis includes division of the cell's nucleus. So when we talk about mitosis, we're not actually talking about the cell dividing. We're talking about the nucleus dividing. And then the very final last stage here is cytokinesis, is literally the cell splitting apart. So the cytoplasm, all of the organelles, everything splits apart completely at cytokinesis. And then you end up with two identical daughter cells. Mitosis can only occur if the cell is large enough and the DNA is undamaged. Cells divide at different rates. The rate of that cell division varies with the need for those types of cells. For example, your skin cells last about 2 weeks, and then they die, they slough off, and they're replaced. Hopefully you know that your skin grows from the inside and out, so you're not making new skin cells on the outside. So the skin that you actually see is a layer of dead skin cells. Your red blood cells, though, live on average for about 4 months. A liver cell lives 300 to 500 days, so around a year, maybe a year and a half. Your intestines, the internal lining, those cells only last 4 to 5 days before they're sloughed off and replaced. And when you think about it, the inside of your intestines has a whole lot more wear and tear, and it's exposed to a lot more things. So those cells have a little bit harder life than, say, a red blood cell. Your intestinal muscles and your other tissues, probably at your age, those are the same cells that originated when you were born, as you were gestating. They probably haven't replicated yet, but they soon will. Some cells are unlikely to divide. So we go back to that cell cycle and we go to those stoplights, those checkpoints. Occasionally what happens is something doesn't replicate correctly. And so the cell will, instead of giving it a green light, it'll give it a red light, and the cell at that point will exit the cell cycle. It'll stop dividing. It won't make a copy of itself because there's something corrupt about it. At that point, the cell enters what we call the GO cycle. The next thing we need to talk about is cell size. So why don't our cells get big? Cell size is limited because they stay more efficient the smaller they are. The volume increases faster than surface area. So if we were to look at 3 similar sized cells, we can see that that ratio of surface area to volume, the ideal size is this medium sized cell. It's big enough that it can support whatever functions that cell needs, but it's also going to allow nutrients to absorb in through diffusion and waste to get out very quickly. This cell, it's a little too small to probably do everything that the cell needs to do. This cell is way too big. It's like Goldilocks. This cell is much too big because the rate of diffusion, those nutrients can't get in. The things that the cell needs in the nucleus can't get in, and the waste products can't get out fast enough. And so the cell will actually either starve or poison itself. So the one in the middle is just right. The surface area has to allow for adequate exchange of materials. That's what keeps the cell size appropriate for its need. One of the largest cells in the human body, we all started off the same size. The human egg is the largest cell. We can actually see it with our naked eye. We don't have to have a microscope to see it. The human egg is the size of a period at the end of a sentence in your textbook. That's how big it is, but it's still efficient enough. So we have red blood cells and we have nerve cells. They're all specific for whatever their function is. Their shape determines their function. And cell growth is going to be coordinated with division. So as the cell grows and it gets ready for division, it's going to change in its size. Cells that have to be large have very unique shapes to enable them to be more efficient, to move things in and out. So think about like a store. If you go into a grocery store that's really, really big and you're trying to find one thing, it's going to take you longer to go in and find it and then get back out. But if it's a small store, you can go in, quickly find what you need and get out. It's the same thing with a cell.